Welcome to Neurofrontiers, a series in which Practical Neurology partners with ReachMD to bring you clinical research and advancements that further clinicians' ability to diagnose and treat neurologic conditions. In this episode, Luka Kulic, MD from Roche, Basel, Switzerland, discusses the investigational BrainShuttle technology.
Dr. Kulic, thank you so much for joining us today. Wondering if you could just start by telling us a little bit about yourself and your role at Roche.
My name is Luka Kulic. I am a neurologist by training, a neuroscientist, and have more than 25 years of experience in the field of Alzheimer's disease therapy and research. My role at Roche is I'm Vice President and Global Head of Early Clinical Development in Neuroscience and Rare Diseases, and have been the early development lead for trontinemab, a BrainShuttle molecule that is currently being investigated in a phase 1b/2a study and has just transitioned also to phase 3.
Please provide an overview of the BrainShuttle technology with an emphasis on its unique method of drug delivery.
The BrainShuttle technology leverages a physiological mechanism that is this so-called transferrin receptor mediated transport mechanism that occurs at the level of the brain capillaries. This is a physiological mechanism that enables transport of, in case of the transferrin receptor, of transferrin and iron across the blood-brain barrier. The BrainShuttle technology leverages this physiological transport mechanism to smuggle in therapeutic molecules across the blood-brain barrier into the brain to target brain pathologies, in this case, amyloid plaques, in case of trontinemab.
Great. Thanks so much. That's a great overview. And how does this technology, the BrainShuttle, compare with other methods of delivering large molecules across the blood-brain barrier?
This is a specific mechanism that leverages a physiological transport mechanism that happens at the level of the brain capillary. It's an active transport mechanism which ensures rapid and enhanced transport of the therapeutic molecule across the blood-brain barrier. And that happens throughout the brain in a very homogeneous manner. That is, I think, also a very important feature. In one of my previous presentations, I showed, for example, a picture where we looked at the distribution of the BrainShuttle molecule as compared to a standard antibody. So a BrainShuttle antibody versus a standard antibody. And what you see is that the access via the brain capillaries, which are everywhere within the brain, results really in this enhanced and homogeneous access to the brain. While, for example, a standard antibody is more restricted to certain areas of the brain. So it does not lead to a homogeneous penetration of the brain tissue.
And trontinemab uses this BrainShuttle technology to improve blood-brain barrier penetration, as you're describing. Mm-hmm. How does this strategy overcome some of the historical delivery limitations that are found with other Alzheimer's disease therapies?
Trontinemab is our lead BrainShuttle program. So the first one actually at Roche, with which we have really established a proof of concept for the whole platform. The difference, and we have studied trontinemab also and compared trontinemab also in preclinical experiments with regards to brain penetration, what we've seen is really that we get more over the blood-brain barrier in a more homogeneous manner, and that we can target brain pathologies like amyloid plaques more effectively and more broadly throughout the brain. And what is also important to note is that with this technology, since we can get more over the blood-brain barrier, we can also use a lower dose to achieve the same or even more than we usually achieve with standard antibodies, which have a much harder actually time to cross the blood-brain barrier. Standard antibody, like gantenerumab, the one that we studied years ago at Roche, their brain penetrance is actually quite low. So it's essentially 1 out of maybe 3,000 molecules that we inject intravenously gets into the brain tissue, while the BrainShuttle technology really ensures that it's several-fold increased. Mm-hmm. And not only increased, but then also it's homogeneous throughout the brain. It's really an important differentiation feature.
Sure. In addition, you just mentioned the ability to use a lower dose. How else might the technology impact efficacy and safety of this therapy as well as others?
We are currently studying the efficacy of trontinemab in the phase 3 program, so Trontier 1 and Trontier 2, so this is currently ongoing. But in terms of target engagement and clearance of amyloid plaques, what we see is very rapid and robust amyloid plaque clearance. This highlights the potential of this technology. But significantly lower doses than what we usually use with standard antibodies. We know today there is really emerging evidence that rapid and robust plaque clearance is necessary for clinical efficacy. So the more patients we get below a certain threshold of amyloid positivity, the earlier this happens, the more likely will the patients benefit. So there's emerging evidence from the field suggesting that. And trontinemab, and this is what we presented also at this conference, achieves this very rapid and robust amyloid plaque clearance. So as an example, after 28 weeks, we saw that 92% of the participants in that study were cleared, so fell below the amyloid positivity threshold of 24 centiloids, which is very fast. What is also important to note is that safety profile, the safety and tolerability profile remains favorable. And I think what has been very important for the field is that the amyloid-related imaging abnormalities, so this is a common adverse event of these anti-amyloid therapies, is actually much lower than what we see with standard antibody approaches. And I think this combination of very rapid and robust amyloid plaque clearance that is likely associated with better clinical outcomes, coupled with an significantly improved safety profile when it comes to ARIA, is something we are very excited about with trontinemab.
Yeah, it's fascinating. Great. Thank you so much. And the presentation you mentioned at this year's Clinical Trials on Alzheimer's Disease meeting included results of the phase 1/2 BrainShuttle AD study. Can you briefly describe the key results and the clinical applications of that study?
Yes, it is a phase 1b/2a study that we started in 21. It is a relatively complex study. It has different parts. And here at CTAD, we gave an update on the first two parts of the study. Part one is the so-called dose finding or dose escalation part. That's a smaller part with also a smaller number of patients. And then there is a part two. What we did there, we essentially expanded cohorts that looked promising based on the amyloid PET reduction, and also the safety profile looked promising in part one. We expanded those cohorts. And that occurred with what we call cohort 3 and cohort 4, so 1.8 and 3.6 milligram per kilogram. And what we presented at CETA was the complete data from part 1 and part 2 of cohort 3 and cohort 4. 92% of participants below the amyloid positivity threshold. 70% were actually very deeply cleared. So they reached levels below 11 centiloids, which is kind of another threshold, which suggests very, very also deep clearance of amyloid. Low ARIA-E incidence. ARIA-E is a form of amyloid-related imaging abnormalities that people look carefully at in these trials. And there we had only one participant in cohort 4. So this is active drug and placebo combined in a 4:1 ratio, but 1 out of 75 who developed ARIA-E, which is a very low incidence overall. So that highlights really this improved safety profile with regards to ARIA.
What are the clinical implications of therapies generally that are delivered with BrainShuttle in terms of AD pathology clearance?
Yeah. So as I said, I mean, there is emerging evidence in the field that a rapid and robust clearance is necessary for efficacy. So the learnings over the past couple of years from the field have been that those trials in which amyloid was lowered, but not like to such an extensive or such a great extent, those trials turn out negative. So that's important to note. So only those trials where patients during the double-blind treatment period, which is usually 18 to 24, 27 months, you need to get the levels of amyloid below the threshold of amyloid positivity in most of the people to see a signal. Those trials that did not achieve that were negative. Those that achieved clearance in a majority of patients were positive. And we have also done at Roche some research into these relationships, including a meta-regression analysis that we actually published 2 years ago, where we looked at the totality of data, trial data from the field that suggests really that in those trials where early clearance was achieved, so at an early time point, so for example, in an 18-month trial after 6 months, if the more people we get at this early time point below the threshold, the larger the efficacy signal is at trial endpoint. So in an 18-month trial, if you see the majority of participants below the threshold at 6 months, that's a good predictor of the clinical efficacy signal at 18 months. And based on this meta-regression, we believe that the higher the percentage of people at this early time point that become amyloid negative, the larger the effect size will be. This is currently based on these meta-regression data. It's not proven. It needs to be proven now in the real experiment, and that is ongoing in the frontier studies. But there is some hope to believe that this improved and early deeper clearance may result in a greater efficacy signal. But of course, apart from efficacy, it's really also the safety profile, because this is a big concern in the field. I think also to some skepticism around these therapies and also to access challenges. That's why I think it's a great advancement that we can essentially clear amyloid with a relatively low ARIA incidence. So that's, I think, another kind of key aspect that needs to be highlighted.
Okay, great. And on that topic, you mentioned the low ARIA incidence you've observed and some of the safety considerations and concerns around that. Are there any other specific safety considerations or delivery limitations that are associated with BrainShuttle?
In our trial, the most relevant or most frequently reported treatment emergent adverse events are infusion-related reactions. So this is something that is not uncommon with biological therapies. What is important to note is that these infusion-related reactions are generally mild to moderate in severity and that they are well manageable with pre-medication. What we do is 30 minutes prior to the infusion, the patients get a steroid injection, one injection, and then they get the infusion. What we know is that infusions that were pre-medicated with corticosteroids, they were associated with infusion-related reactions only in 6 to 7% of cases, as compared to more than 30% of infusions that were given without any pre-medication. So that highlights that pre-medication works and that this is a viable and reasonable mitigation strategy.
And the results we've talked about and the results you presented at CTAD have focused on the role of BrainShuttle for delivery of Alzheimer's disease therapies. What are some of the other potential applications for BrainShuttle in other fields of neurology?
Alzheimer's disease is one of the indications we're exploring, and trontinemab is the most advanced program in a larger indication like Alzheimer's disease, in a sporadic disease like Alzheimer's disease that we are developing now in phase 3. We are generally excited about this technology, and are also in our portfolio at Roche exploring other shuttle molecules and also for other indications. For example, there is one program that is currently in phase 1 in multiple sclerosis, where we are using the same technology to deliver an antibody that targets B cells, so a CD20 antibody over the blood, to essentially get this antibody over the blood-brain barrier, leveraging the BrainShuttle technology with the idea to target really B cells, which are known to play a pathological role in MS, in compartments that are not easily reached by standard anti-CD20 antibodies. So this is one of the programs that is currently in the clinic. But of course, in our earlier pipeline, we're exploring multiple BrainShuttle molecules for multiple different indications.
Thinking through some of the next steps for BrainShuttle, you mentioned the frontier studies, and can you just give us an idea as to the future directions for the BrainShuttle technology?
So as I said, I think Alzheimer's disease and targeting amyloid is, I think, a great start. The proof of concept that we have established now in the phase 1b/2a study highlights the potential of this technology for a wide range of neurological indications, where the blood-brain barrier is really essentially limiting. The blood-brain barrier restricts the access of all kinds of molecules, especially large molecules, to the brain. That opens up new avenues for targeting different pathologies in the brain. As I mentioned, the CD20 target in MS, but there are other also misfolded proteins in Parkinson's disease. There are also other proteins than beta amyloid in Alzheimer's disease, like tau, that really highlights the potential to reach these brain targets more efficiently and more broadly, which will hopefully result in a much better efficacy than what we see today with our therapies that actually are limited by the fact that there is the blood-brain barrier, which limits the access of the vast majority of these molecules that we inject systemically.
One last question for you. What should physicians tell their patients if a patient comes and asks about the technology after seeing it in the news, for example?
It's still an investigational therapy. It's not approved. It's not a marketed product. But it's currently being investigated in phase 3 in the Frontier studies. There is obviously the possibility, right, to participate in clinical trials if patients are interested, and of course, if they are eligible, of course, for these trials. So that is currently the option. This is, I think, a promising therapy, however, still an investigational stage where we need to really establish properly in a phase 3 trial the efficacy and safety profile.
Thank you so much for joining us. Really appreciate you sharing this with our listeners.
You're welcome.
Thank you to Dr. Kulic for his insights, and thanks to you, our listeners. NeuroFrontiers is brought to you by the editors of Practical Neurology. Please visit practicalneurology.com for more podcasts in the field of neurology.